Now, the disorders present in pyrimidine metabolism is very simple. So, really, there's only one, which is orotic aciduria. So, orotic aciduria is caused when there might be increase in production of orotic acid or the excretion of high amount of orotic acid. And when there's high amount or higher production, there will be a decrease in formation of RNA and DNA, which is a very, very bad thing and can cause so many defects. So now in orotic aciduria, it's due to the deficiency of UMP synthase. So UMP synthase is a bifunctional multi-enzyme which contains two enzymes, which is the first enzyme is orotate phosphoribosyltransferase transferase, and OMP, which is orotic monophosphate, Uridine monophosphate, or Decarboxylase. So these two enzymes, when they are deficient, they can cause orotic aciduria. But you have the type 1 and type 2. When you have deficiency of these two enzymes, you have type 1 orotic aciduria. But when you have deficiency of OMP decarboxylase, you have type 2 orotic aciduria. So one thing you have to know about type 1 orotic aciduria, when you have deficiency of the two enzymes, what happens? There will be increase in orotic acid in the kidney, which can cause kidney stones. The kidney stones will be will contain high amount of orotic acid. Don't forget, remember, when there's high amount of, dihydroorotate uses is being oxidized. Dihydroorotate is being oxidized to orotate or orotic acid. So if the enzyme, if there's no orotate phosphoribosyltransferase transferase to form OMP, which means there will be increased amount of orotic acid or orotate, and this will be present in the kidney, causing kidney stones, increase in orotic acid. The same time, there will be severe anemia, usually megaloblastic anemia. So there will be megaloblastic anemia here. Why? Because there is absence of folate. So folate is important in the reaction between DUMP to DTMP. So there will not be folate. What will happen? Megaloblastic anemia will happen. During the same issue now, there will be leukopenia. So which means there will be a decrease in amount of leukocyte present. So white blood cell will be low. So immediately white blood cell will, the patient or the individual will have what we call immune deficiency. So there will be lot immune deficiency, and most of them do not survive, they die. In type 2 orotic aciduria, when there is absence of absence of OMP decarboxylase, what happens? This one is also the same thing. What happens here? There will not be high amount of orotic acid excretion. There will be high amount of orotic acid secretion, and also there will be severe anemia too. Anemia will also be present here. So, but in this one, there's more a middle case in this reaction. There is more middle case in this reaction here than in the previous orotic aciduria type 1. So generally, what happens to patients that have type 1 and type 2, like we all said, in type 1 and type 2, there's usually presence of kidney stones. There's presence of kidney stones. There's retarded growth. There will not be growth because there's no DNA and RNA fibroblast growth factor to make growth possible. There will not be severe anemia, either megaloblastic or hypochromic anemia. Also, there will be leukopenia, like we said, there will be decrease in white blood cell count. So the only way we can treat it by administering the patient with uridine or cytidine. So when you give uridine or cytidine, what will happen? There will be a kinase reaction to convert uridine or cytidine to UMP to CMP. So UMP and CMP, what happen? They inhibit CPS II. That is it. So when you administer uridine, and you administer cytidine, what happens? They will be phosphorylated by nucleoside monophosphate kinase to UMP and CMP. So when they are phosphorylated, what happens? They will inhibit CPS II. What will happen? There will be decrease in orotic acid formation. So this is the clinical correlation of pyrimidine metabolism. So next is the regulation of pyrimidine biosynthesis or pyrimidine metabolism. In this reaction, we are only regulating allosterically. Unlike others, we regulate dietarily and hormonally. Here we are regulating allosterically. In the allosteric regulation, we'll be looking at some key enzymes, the CPS II, Carbamoyl phosphate synthetase 2, used in the cytosol in pyrimidine, in contrast to CPS 1 using urea cycle. So now our CPS 2 is very, very important, the important allosteric enzyme, which is activated by ATP. So ATP activates CPS 2 to continue the reaction, but they are also inhibited by UDP, which is uridine diphosphate, UTP, DUTP, and CTP. So all these all these products of pyrimidine metabolism inhibit what? CPS2. Only ATP activates it. The next one is aspartate transcarbomoylase. Aspartate transcarbomoylase can be activated by ATP, same thing, and inhibited by CTP. Don't forget. So it's a key allosteric enzyme used in this reaction. So the next we will be talking about here is now the whole pyrimidine metabolism can be activated generally by they can be activated by PRPP. PRPP can activate them 100%, and they can be also be activated by ATP, all of them. So all of them can be activated by ATP and PRPP. So when you increase when ribose 5-phosphate is converted to PRPP, so PRPP continues the formation of pyrimidine bases, but their product, which is CTP, UTP, and TTP, DTTP, usually inhibit the reaction. So the product inhibits while PRPP and ATP what activate the reaction. So now in the UMP synthase, now in the UMP UMP synthase, which is the functional multi-enzyme here, what is their regulation? The first one is OMP decarboxylase. OMP decarboxylase, which is orotidine monophosphate decarboxylase. But remember, what do we do when we form OMP? What do? We now use decarboxylase enzyme. We decarboxylated it, removing CO2 to form UMP. So UMP, we inhibit the reaction. So UMP is the allosteric inhibitor of what? OMP what? Decarboxylase. Don't forget. So it prevents them to a lesser degree. It's not inhibited by what? CMP. So CMP is also inhibited to a lesser degree, but 100% is inhibited by the product, which is what? UMP. So UMP is inhibiting it. Don't forget it. So since we have talked about the allosteric regulation of what pyrimidine metabolism,